Let us take a hypothetical situation where we have our bone, let us say we are taking an object in our hand. This is the femur bone, this is the forearm bone. We have this muscle which is connecting this bone to the femur bone, and for every bone, sorry, every muscle there is a antagonist muscle. So, in this case this is, let us say this is a biceps, here is the triceps, excuse me for my poor drawing. Okay. Now, this muscle is connected to our spinal cord, which we have seen it in the earlier class. Let us say it is connecting to the spinal cord, let us say that is a spinal cord. In the spinal cord, this muscle is connected to a motor neuron. The last class we saw that motor neuron, right, motor neuron, and is what it is going to, let us say it is connecting this. So, a muscle needs electrical impulses from the motor neuron. Whenever the motor neurons fires, then the muscle contracts. Muscle can only contract, it cannot elongate, it cannot generate force when it is elongating, right. So, this connection from motor neuron to the muscle is called the efferent pathway. Okay. Now, the muscle when it is contracting, then the two bones come together, the length of the muscle changes. If you want to control these forces generated by the muscle, then there should be some way of now monitoring the muscle length and force, right. Then only the closed circuit, a control circuit will be established. So, somehow we need some mechanism to monitor the length of the muscle and the force of the muscle. Let us say we have some sensors over here, and the sensor is attached to the motor neuron. Okay. This is called the afferent. Afferent is anything that sensor is going to the central nervous system. This is a CNS, part of the CNS we saw, right. Efferent is anything from CNS to the effectors outside, that is the afferent and efferent. This again one terminology which you may have to now get used to it. Now, the question is now how is, what kind of sensors will be here in the muscle which is measuring the length, and we are going to see that it is not just measuring the length, but also many other things. Okay. So, with the engineering background we have seen a lot of sensors, right. So, what sensor will measure the length? Can you name some sensors that measures the length change? LVDT, very good. Any other sensors? Change in length, potentiometer, that is the rotation. Linear potentiometers are also there here, very good. Okay. Strain gauges, very good. Capacitive sensors, okay, all right. So, we have so many sensors that can measure the length changes. So, but what changes, what sensor is in our muscle? Can you imagine a LVDT in the muscle, or capacitor or potentiometer is there? What sensor is there in the muscle? Which one? Piezoresistive is there? Okay, very good. All right, let us see what kind of sensor, what nature has designed a sensor in our muscle, okay, and how it is working is what we are going to talk about it. Okay. So, before we talk about, there is one more concept I need to clarify. It is So, this muscle is with only one motor neuron in between, it is called a monosynaptic synaptic motor neuron, right. There is only one motor neuron connecting the receptors to the muscle. Okay. When this muscle is contracting, imagine this muscle also is contracting, what happens? That is a triceps, this is a triceps, right. Triceps also is contracting, then what will happen? No, the hand will not move at all. So, when this is contracting, this has to relax, then only eventually you can contract it. So, somehow this muscle has to be told that no, just relax. So, how will that happen is there is a polysynaptic muscle, polysynaptic neurons are there. From here there is a interneuron that is inhibitory nature, from here it will go to this is again efferent. Okay. So, because there is an interneuron which is inhibitory nature, whenever this motor neuron is firing, this will stop this motor neuron firing, therefore, this muscle is in relaxing condition. So, that this muscle bicep is actually contracting, the entire musculoskeletal system is actually reducing the length, this is reducing the length. Okay. So, monosynaptic motor neuron and polysynaptic motor neuron. Is very important in understanding how the muscle is working. Now, how is the system tells the triceps in this case to relax? Okay, that is a question. Then before we see that more details, there is one more set of terminologies I wanted to introduce. It is called the intrafusal muscles. The muscle is usually called the intrafusal muscle, intrafusal muscles and extrafusal muscles. The sensors we are talking about, which is measuring the length, itself is made up of muscles. Okay, it is not made up of piezoelectric or now potentiometer, it is made up of muscles. So, the muscle is embedded inside the muscle. The embedded muscle is called the intrafusal muscle. The outside muscle is called the extrafusal muscles. This distinction also is very important. So, again to recap, we have seen many terminologies. This terminologies are very important to understand the rest of the things. Afferent is going from receptors to CNS, efferent is from CNS to the effectors. Okay. And then monosynaptics and polysynaptics. Monosynaptics is one and single motor neuron, which is reflexive actions. Polysynaptics is usually multiple motor neurons in maybe inhibitory or intrafusal, extrafusal. Intrafusal is the muscle embedded inside the muscle, extrafusal is the outside muscle, and then we saw kinesthesia and the proprioceptions. Now, let us see how this intrafusal muscle is helping to get the length sensed. So, this muscle what you are seeing is the extrafusal muscle. Okay. Inside you can see the small rectangle drawn over here, you can see the small muscle in there shown in white, that itself is a muscle, and it is a different muscles. There are different kinds of the muscles which we will see it now later. We will not see the different kinds of muscles in this course in detail, whereas in the biomechanics course we study the different type of the muscles in detail, but we will just now quickly overview different kinds of muscle. So, inside the muscle it is a intrafusal muscle that is going to act as a sensor for us, okay, and that is measuring the length, whereas there is another organ you can see that this is the Golgi tendon organ, it is called the GTO, that is measuring the force, the tension which is felt by the muscle, whereas this muscle spindle which is highlighted in a rectangle, that is measuring the length. How it is measuring the length, how Golgi tendon organ is measuring the force is what we are going to see. Now let us say that I have this as a muscle, our spindle organ, this is called the muscle spindle, right, muscle spindle is embedded in the bigger muscle. This is the extrafusal muscle, fusal muscle, and this is the spindle organ which is itself is a intrafusal muscle we saw. So, this muscle spindle there may be more than one spindle in the muscle. Here at the end this is Golgi tendon organ, this also, this side also. Okay. So, you know the tendon is what is connecting the muscle to the bone. Okay. In between the bone and the muscle, this is the GTO which is connected. Okay. This is measuring the tension. If you see this muscle spindle are connected to the muscle in parallel, whereas GTO is connected to the muscle in series. Okay. So, when the muscle is elongated, let us say I am let us say this is an elongated muscle, you can see that this spindle organs also gets elongated. Okay. So, in a parallel arrangement, when the muscle is getting elongated, the spindle also get elongated. Okay. Whereas the GTO gets strengthened, whereas when the muscles get strengthened, the same muscle whereas the GTO gets elongated. So, you can see that in this the GTO which is in series gets opposite of what the muscle is actually doing. The GTO gets elongated when the muscle is shrinking, whereas the muscle spindle gets elongated when the main muscle is elongating. Okay. This arrangement is again is very important. I am going to show you how it is. Now, how is this muscle spindle helping us to measure the length? So, I am going to focus only on the muscle spindle. This is a muscle spindle alone which is embedded in the main. Inside the muscle spindle there are two organs. This is called the again let me put a oops. Oh, yeah, this color is there, yeah. Yeah. So, this is the muscle spindle has bag, nuclear bag it is called. This is the nuclear chain, nuclear bag, nuclear chain. Okay. And the nuclear bag, let us say this nuclear chain is connected to the CNS. This is the afferent, is not it? Afferent, it is one particular type of fiber called 2. Okay. And the nuclear chain also, nuclear bag also is connected to okay, nuclear bag, there is also is connected to, it is called the 1A. Okay. The 1A which is slightly thicker, I am going to write it as thicker one, and there is also connection from the nuclear bag. So, 1A has connection from, this is thicker actually, first of all thicker fiber, nerve fiber. Thicker nerve fiber means quicker conduction when compared to the number 2 type of nerve fiber. Okay. So, this 1A gets input from both bag and chain, and whereas the 2 gets input only from chain. Okay. So, this nuclear bag and the nuclear chain, these are all receptors, just like the receptors what we have seen in the tactile receptor, like Pacinian or Ruffinian, 4 type of receptors we have seen. So, this is again like a receptors. The nuclear chain is like a rapidly adapting receptors. Okay. So, I am going to say this is a whereas this one, the nuclear chain is like a slow adaptive receptor. Okay. As you know, the rapidly adaptive receptors, they will give input, or they will give neural firing only when there is a change, right, whereas the slow adaptive will give firing proportional to length. So, even if it is constant, it will continue firing, whereas nuclear bag will fire only when there is a change, and when there is no change it will not fire. That is a rapidly adaptive receptors we have seen earlier. But if you look at this 1A, which is getting input from both a bag as well as the chain. Okay. So, suppose if you measure this as if you take this as a length measure, this can be considered as measuring the velocity. So, the nuclear chain and bag together measures the velocity of the stretch. Okay. So, the spindle itself, measures not only velocity, not only length, but also velocity. That is a take home message I am deriving it. Okay. Let me write it as velocity and length, it is what measuring it. These are all going to the CNS. Okay. Now, the question is, why does it require velocity and length? Why not just length is good enough? Okay. That we will take it up later. Now, we will finish off how it is actually measuring it. Okay. Okay. So, I hope this part, the muscle spindle is clear. Let me go to the other parts of the muscle. Okay. Here in this slide, I want you to be now familiar with all the terminologies used. Extraphyseal muscle fibers, right? Alpha motor neuron and is the motor neuron coming from the motor neuron. So, essentially let me go back. Okay. This motor neurons can be alpha motor neuron or gamma motor neuron. We are going to talk about 2 different things. I will explain and why we need a gamma motor neuron and in a few moments. Okay. Golgi tendon organs, you have seen it. Okay. So, the proprioceptors or the kinesthesia is coming from the response to the muscle spindles, Golgi tendon organs, and there are joint receptors also. There are joint receptors which are found in the ligaments and the capsules all the membranes which is around the joints, and then cutaneous receptors. So, the tactile receptors which we have studied earlier, that also gives input to the proprioceptions. Okay. Do you remember which receptor is responsible for stretch? Not Pacinian, Ruffini endings are actually responsible for stretch. So, when you move the hands, the skin, some part of the skin is getting stretched, and that could be actually useful for proprioception. Okay. So, not only muscle spindle, but also Golgi tendon organs and the joint receptors, cutaneous receptors. Okay. So, this is spindle organs we are looking at. Yeah, okay. Intrafusal muscles, I am going to, yes. So, in this picture, this is a muscle we are talking about. Let me, yeah. So, can you see the mouse cursor moving? Yeah, this is a muscle we are talking about. In this, there is a spindle, and that in the spindle you can see that there is a nuclear bag and a nuclear chain. Nuclear bag and nuclear chain, you can see that it is a 1A is coming out, 1A is going to the CNS, that is a afferent. 1A is a afferent which is coming from both nuclear chain and the nuclear bag. Okay. Whereas 2, okay, so this is actually zoomed in over here. In the zoomed version, you can see that there is a nuclear bag and there is a nuclear chain, and 1A is coming from both nuclear bag and nuclear chain, 2 is coming from only the nuclear chain. Okay. And here you can see that there are monosynaptic motor neurons, there are polysynaptic motor neurons. Okay. And this is a Golgi tendon organ. From Golgi tendon organ, there is 1B going, that is again 1B is a naming of the fiber, nervous fiber. Okay. Depending upon the size, depending upon how fast it can conduct the signals, it is all named 1A, 1B, right. And this alpha is coming from motor neuron, which is the efferent. Efferent is stimulating the extrafusal muscles. Okay. And this is gamma, the gamma stimulating this, the nuclear bag and nuclear chain. I told you that the sensors themselves are made up of muscles, right. So, the muscle means we can stimulate it and then we can contract it. Why do we need to contract? I will tell you where it is needed, but you can imagine that there is a requirement, and that is coming as a gamma, okay. And this is a alpha, this is a gamma. Similarly, the motor neurons can be either for alpha or gamma, okay. That is the requirement. Now, let me go back and then emphasize on why we need a gamma. It is called the gamma drive. Okay. So, now let me again draw this nuclear bag and nuclear chain. Let us say this is a whole spindle we are talking about. So, the gamma drive it is coming to so the only the end of the nuclear bag and the nuclear chain are muscles. The center is the receptor. Okay. So, this is the gamma drive we are talking about, gamma is coming. Okay. Similarly, here also and this also gamma. So, what happens when you stimulate the muscle? It contracts, correct. So, this part of the muscle alone is going to contract. Similarly, this part of the muscle alone is going to contract. When this contracts, what happens? This center part is going to stretch, is not it? So, it is going to stretch it. Stretch means the receptor is increasing its firing because the length is increasing, right. Similarly, this also is going to contract and this also is going to contract and the center part alone is going to elongate. It means it is again it is going to increase the length. So, what happens is let me go back to the initial one. Let us take this picture. So, when there is a length change, immediately this muscle spindles are going to start firing and immediately this motor neurons are going to ask the muscle to relax or whatever it is. Suppose if you want to maintain the position, what will happen, or if you want to if there is a disturbance, this is a position of the hand, there is a disturbance of the position, some object has fallen in it, immediately the muscle has to act. How does what will happen? Because the length of the muscle is changing, the spindle sends the information to the motor neuron and then motor neuron immediately asks to contract, then it comes back to the initial position. Okay. This is called the stretch reflex, stretch reflex. That is automatic, it is a monosynaptic, that is the shortest reaction possible. Reflex is something that is fastest possible, right. Now, suppose if we want to voluntarily stretch your hand, what happens? The muscle spindle again is going to happen, it is going to increase. So, you are going to say this way you are going to stretch it, this way you are going to stretch it. So, the muscle spindle is increasing in its length. When it is increasing in the length, again this motor neuron is going to fire it and then it is going to ask the muscle to contract it, right. So, you will never be able to stretch if this is the case. If the stretch reflex is present all the time, if the stretch reflex is present all the time, you will never be able to voluntarily move anything. Okay. So, stretch reflex is involuntary, but whenever there is a voluntary movement necessary. So, what the technique the nature our brain uses is that it sends it asks the gamma motor neuron also to send signals to the intrafusal muscles. Okay. So, that is where we are here. So, now the gamma motor neurons also, gamma motor drive also is working. When it is working, what happens is this is getting stretched and this is also getting stretched. Okay. So, when this is getting stretched, the center part is actually maintaining its same length in the voluntary conditions. So, this is only for the voluntary conditions. Whenever there is a voluntary movement, this gamma drive is working, gamma motor neuron is sending the signals to this intrafusal muscles and this center portion is maintaining its length. So, there is the CNS does not see any change at all, any change in the muscle length. Therefore, the motor neuron, alpha motor neuron is not going to fire. That is because these are all connected, we know. We have seen that this is all connected to, what is it? This is 1A. Let me put it at this one. Okay, green is the green working. This is a 1A and this is all 2A. We have seen it, right. So, the center portion, it is connected, all these fibers 1A and 2A, sorry 2, you have seen, you have to observe that this is connected to only the center of it. Okay, that is a receptor, main receptor. The main receptor will not change in case of the voluntary movement. That is because of the gamma drive. Gamma will be proportional to how much of voluntary you want to do it. This gamma motor neuron, which is in the spinal cord, will send signal proportional to whatever is necessary to make sure that these receptors are not firing, so that alpha motor neuron does not fire at all. Okay. So, the concept of coactivation, this is called the coactivation of alpha and gamma motor neurons together. So, only with a coactivation of alpha and gamma motor neurons you can voluntarily move, otherwise you can't move at all. The stretch reflex will be all the time there. Okay. That is a concept I wanted to take. Okay. Now, let us look at this picture again. You can see that this is a gamma. Can you see this? Okay. You can see that this is a gamma drive coming up. Okay. That is to the end of the nuclear bag and nuclear chain, and this is a 1A afferent. There is also dorsal root ganglion which is over here. This we have seen it in the last class. Okay. And then from the brain cortex there is a descending motor traffic which is coming over here, but this is the alpha motor neuron over here. This is a monosynaptic, this is a polysynaptic, this is a gamma motor neuron. This is a ventral root, there is a dorsal root. Okay. Is this picture clear? Right. We will move on to the next one. Now, Golgi tendon organs. Let us look at Golgi tendon organs in little detail. So, tendons are very tough fibers, right. Inside the fibers, okay, there are afferent neurons which is embedded over here. Okay. So, these fibers are made up of collagen fibers which are very tough over here. So, you can see that the blue colored are the sensory neurons. So, when the organ is getting stretched, you can see that the sensory neurons are getting compressed. Depending upon how much it is compressed, it will send out the firings. That will be a measure of how much force the muscle is actually experiencing or applying. Okay. So, spindle we have seen, Golgi tendon organs we have seen, cutaneous receptor also is contributing to the proprioception we have seen. Stretching of the skin, limited role in proprioception. Their main role is tactile sensation, but it has some secondary role in proprioception as well. Also, knee kinesthesia no effect at all. Mouth, hands and feet proprioception significantly reduced by kinesthesia, whereas in the knee kinesthesia does not have any effect at all. whereas in the mouth, hand and foot, when you have a anesthesia, the proprioception significantly reduced. It means, and there are joints and or cutaneous receptors which is contributing to the proprioception significantly. Ruffini endings, since they are slow adaptive, population of the neurons responding simultaneously, that has very significant input to the cutaneous receptors. Okay. So, let me okay. So, I will in the next class we will talk about the what are the different type of the movements and how kinesthesia, basically we are going to talk about this, some psychophysical measurement of the kinesthesia. There are more concepts such as the motor unit and all those things we will see in the next class. Now, I will take questions from today's class. If you have any questions, we will discuss.
Measuring that force.
How is it measuring force? Okay. So, you can see that there are collagen fibers. Basically, tendon is nothing but fibers, tough fibers. The fibers are made up of the collagen fibers. So, this sensory neurons, they are intervened with the fibers. So, when there is a tension, all the fibers are compressing this sensory neurons. So, depending upon the tension, these fibers are compressing the sensory neurons. Okay. So, the sensory neurons fires depending upon how much the fibers are compressing it. That is the way it is measuring the force. Okay. Any other doubts? Let us go back to the first one. Now, the kinesthetic receptor, let us look at it. Spindle stretch receptors, we have looked at in some details. Spindle tension receptors, that is nothing but the Golgi tendon receptors. So, nuclear bag and nuclear chain fibers, we have seen it. Okay. So, dynamic bag fibers, static bag fibers, that we have again seen it. Bag has two components, dynamic and okay. Now, one of the questions I asked, probably we can clarify it now. Why do the muscles need length as well as the velocity? Okay. Can anybody guess why do we need length as well as the velocity? Okay.
Sometimes we need reflexes.
Sometimes we need reflexes. Okay.
To go to one position to another position, we need not record only length, we need velocity also to go to that another position.
Yeah, we need velocity also, right? So, yeah, the answer is this. If you do not want to overshoot it, if you take any control systems, okay, I want to move from this point to this point, but I can move it without overshooting it. So, if you have only length based control, then what happens is going to slightly overshoot, and then finally I will settle in the point, but that is not happening in the hands. When we have the velocity without overshooting, we can go there to the final point. Okay. This is called the PID controller, you have all seen it, right. Here we are talking about proportional to the length as well as the derivative of the length. So, proportional plus derivative is a concept, proportional to proportional plus derivative. So, I can explain this in a simple example. Let us take one pendulum. Let us say the pendulum has m over here, and that is at theta, and it has to come to the resting position without overshooting. Okay. Suppose if we have a torque at this point which is proportional to the theta, then it will overshoot. If there is no damping, it will infinitely overshoot, I mean it will oscillate it. If there is a damping, then there will be damped oscillation, and finally it will settle there. But instead of a torque proportional to now theta plus some constant times theta dot, then we can show that this is actually settles without overshooting. Okay. You know this our simple pendulum equation is theta double dot plus g by L into theta is equal to 0, that is without the proportional, whereas here this is with proportional to theta, torque proportional, restoring torque proportional to theta alone. So, the same equation we can write theta dot plus g by L theta plus g by L k times theta dot. We can show that in case of the proportional to theta plus k times theta dot. So, this equation will make sure that it settles without overshooting, and this is what our muscles are using it. For this we need the velocity also. So, theta dot is nothing but c. Okay, sorry, here it is only the theta dot. We need the velocity also. Okay.